Hello, good morning. Today we will understand the lipids. As we know that lipid is the most important component found in human bodies and most of the sources which are producing. For example, we are eating the food materials of cereals, oilseed and pulses. Out of them, those which are rich in oil or fat, we are called as, or we tell them as a oil seeds. And these oil seeds are rich in lipids or fats. There are different functions of lipids. We will understand in this lecture classification of lipid, structure of lipids, classification of fatty acids, classification of their component on the basis of various carbon chain, and lastly we will understand what is the importance and functions in human body. As we know that these lipids comes the word called as lipos. The fat and their derivatives are also known as lipid. The Greek words lipos means what? Lipos means fat. These lipids or this fat or oil, the principal component found in that is fatty acid. Now, before going to a whole classification of lipids or understanding of lipid, we should know who has invented this word. So, the term lipid was first coined by this scientist, that is known name it called as Bloor, Bloor, in 1943. And during that period they have defined certain these compound is naturally occurring which are insoluble in water and soluble in organic solvent. Now, what are the organic solvent? Benzene, chloroform, ether, petroleum ether. These are comes under the organic solvent. Now, what is the functions? In general, the lipid work as a fuel molecules. Why it is called as fuel molecules? Because it comes under primary metabolites. And what are the meaning of the primary metabolite? In previous lecture, I have already told you primary metabolites that gives you energy, and lipid also gives you double energy than the carbohydrate and proteins. Now, lipids serve as a fuel molecules. Therefore, we are telling. Similarly, another word is signal molecules. How you can say that these are the signal molecules? Our brain, the synaptic gap between these two neurons, these anything which are happening in our body, that is due to the signal molecules, and that is also one of the component of lipids. Similarly, in plant cell and animal cell, there are a membrane bound nucleus or membrane bound organelles are there. These are made up of these lipid. Similarly, if we see the human bodies, most of the hormones or these hormones precursor or indirect effect of this lipid based on this. So, whatever the hormonal regulation is maintained through the cholesterol, and cholesterol is also a part of lipids. So, chemically, how you can define lipids? Oils. These are the terms. Oil and fats. Generally, oil are liquid at room temperature. Fat are solid at room temperature. So, lipid describes chemically we write. There are certain chemical constitution found in that CH3, CH2, 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 COOH, n number of that CH2N, 2N minus 1. That I will teach you later on. These fatty acid substances are highly concentrated energy storage. How it can store this energy? Because it has a double bond system in that. Fatty acid also classified, that I will teach you, unsaturated fatty acid and saturated fatty. Unsaturated means double bond system found that, and saturated means no double bond system. If you define chemically what is lipid, then you have to tell they are ester of long chain fatty acid and alcohol. This is all about the introductory parts of the lipid. Now, how it is occurrence and what is the importance in day-to-day lives in various animals too. For example, they are widely distributed throughout the plant and animal kingdom. As I said, they are present in seeds, nuts, fruits, all living beings, cell organelles, cell wall bonds, materials. They are present in adipose tissues of animals, bone marrows and nervous tissues too. Why this picture is giving? Because these two animals can survive their life up to a minus temperature, means cold environment they can survive. Why? Because they having a characteristic of lipid composition in their cell wall. Similarly, now I will give you a simple cell membrane structure. This cell membrane structure is composed of, as we know that cell membrane, any cell membrane is made up of, these are the components, hydrophilic head and hydrophobic head there. So, in integral protein are also there. These integral protein, single transmembrane helix and another multiple trans helix is like this coiling. Okay. Similarly, peripheral protein, that is at end of that, I will show you here the structure of these. You will find peripheral proteins. One of the component of cell wall or cell membrane, these are the glycolipids. Phospholipid is the polar head. Remember this word, polar head, phospholipid. Peripheral protein covalently linked to this lipid. Thereby, here you will find a polar head, and outside there are certain glycoprotein attached with the oligosaccharides. So, why the cell membrane is composed of this component or lipid bilayer? I will teach you later on in the cell membrane. Now, where it is found and how it, what is the composition or chemical found in that. So, lipid micelles is structured like this, water molecules and no water molecules in between that. So, the lipid bilayer is composed of these, and hydrophilic head at upper positions interact with the waters and hydrophilic heads. Now, the two terminology comes, hydrophobic and hydrophilic. Philic means it love water. Phobic means it hate waters. So, hydrophobic tails interact with each other. So, the movement of the protein and other molecules can possible or transmembrane of this. Okay. Now, classification of lipids, we will get on. Based on these biological functions, lipid can be classified into two parts. One is storage lipid, another is structural lipids. Now, how these storage lipid? So, these are the principal component or principally stored form or in the form of energy, and structurally, as I said in the previous, in very simple form, I have given only a structure of that. Structural lipid, the major structural element of the biological membrane, as I said previously, and now first two function, storage, another is structural. Now, classification, general classification of lipids, then you have to remember simple, complex and derived, these terms. First is simple. Simple lipid is classified into three are the example, oil and fats. Mostly they are composed of triglycerides. Similarly, wax ester. Wax esters means you will find in honey wax, that is also one of the component of simple carbon wax. Then sterol, there are a major classification of sterol also that I will teach you later on. Here is complex. Complex lipid is classified into two. One is phospholipid, another is glycolipid. Why it is called complex? Because it is attached with the lipid component with the glucose or other non-lipid component. If phosphorus is attached with the lipid, then it is called phospholipids. This is general and it is found in the, as I said, in the cell membrane. Now phospholipid is also classified into two groups. One is glycerophospholipid, another is sphingolipids. Glycerophospholipid is also classified into three: PC, PE and PI. Now how you will pronounce this? Phosphatidylcholine means PC. That can also be asked in the full form. Phosphatidyl ethanolamine. This ethanolamine, that is PE. Another is phosphatidyl inositol. These are the components found in the phospholipid and it classified as a complex lipid. Similarly, these complex lipid, glycerophospholipid, we have completed. Now sphingolipid, there are two things, sphingomyelin and ceramides. We are dealing with the lipid classification, remember this. So glycolipid, glyco and lipid, two moiety, one is sugar moiety is attached with the lipid, cerebroside and ganglioside. Now another terms and which is very important in every living beings and every organism, these are the derived lipids. How it can says that derived, I will explain you by text. One is fatty acids, another is sterols. Here is sterol ester is there, so don't misinterpret that word. Sterols, monoglyceride, diglyceride, triglyceride, there are MGDG, DGDG, that I will teach you later on. So the classification of lipid, that is simple lipid, complex lipid and derived lipid. Now complex lipid is also classified into two, one phospholipid and glycolipid, then it further also classified the phospholipid. So these are the classification of lipid. Now same thing I will explain in the text form. First, lipid are classified into, as I said, simple lipid, complex lipid, derived lipid and miscellaneous lipid. Certain literature you will find. Now simple lipid. What is the definition of simple lipid? How you can write what mean by simple lipid? They are ester of fatty acid with alcohol, OH. They are three types: waxes, sterolesterol and triacylglycerol. Compound lipid, this lipid also ester of fatty acid with alcohol with additional group such as phosphate, nitrogenous base. They are again divided into three types: phospholipid, glycerophospholipid, sphingolipid, sphingophospholipid. These are the derived lipids. These are obtained from the hydrolysis of simple and complex lipid. Now hydrolysis means it is degrade, lysis or breakdown. So these lipids contain glycerol and alcohols, and this class lipid includes steroid hormones, ketone bodies, hydrocarbons, fatty acids, fatty acid alcohol, monoacylglycerol, diacylglycerol. These are the class, and miscellaneous lipid are there, polyketide, that I will teach you later on. These include compound which contain a characteristic of lipid. Now squalene, terpenoids and most of the pigments which are made up of these complex or derived or miscellaneous lipid, for example carotenoids. So these are the, now we have to understand, wherever you will find oil, fat or fatty acids, first of all understand the chemical structure of that, how it look like. So there are two moieties, triacylglycerol, tri means three, glycerol, OL, OH. Now, the structure is glycerol CH2OH, CHOH, CH2OH, 3 carbon sugar. This is attached with the R1, R2, COO, R1, R2, R3. Now, what mean by R1? R1 means the CH2 n times or 16 times with the CH3 at here is COOH. So, this OH here is H is removed. So, triacyl, acyl means COOR1, R1, R2, R3. Similar, CH2 16 CH3, CH2 16 CH3, CH2 16 CH3, 16 into, so 16 carbon these 1, 17, 18. Okay. Similarly, triacyl glycerol are non-polar complex lipid. Non-polar complex lipid. They serve as a energy source or energy reserve. These lipids are triester. Why triester? As I said, 3 fatty acid esterified to 3 hydroxyl group. This hydroxyl group is esterified 3 hydroxyl group of glycerol. Now, fats, if you in general terms, you will find fats are to one animal origin or plant origin. Here is fats from animals are triglycerols. They are solid at room temperature. As I said, what is the difference between fat and oil? Oil mostly from the plant species. So, they are also triglycerides, but they are liquid at room temperature. So, structure of triglycerides I will explain in detail with OCO, R1, R2, R3. These are the chains. So, triacyl glycerol, this esterified that contain the same kind of fatty acid in all 3 positions, 1, 2, 3, called simple triacyl glycerol. Otherwise, that mixed TAG is also possible. Here you will find icosanoic acid 20:1. Delta means double bond system at 1. See, this is MUFA, mono unsaturated fatty acid. Similarly, oleic acid is also a MUFA 18:1, 1 double bond system at delta 9 positions. So, these R1, R2 and R3. Uric acid, that is 20:1 at delta 3 position at 13 positions, that is also uric acid. So, these are also example of triacyl glycerol. This is found in the different compositions in different oilseeds as well as non-oilseed crop also contain less than 1 to 10% of oil too. Okay. So, to understand storage lipid we have completed. Structural lipid, how it's look like, same storage lipid, that as I said neutral lipid, triacyl glycerol, this glycerol plus 3 fatty acid 1, 2, 3. Now, glycerolic 3 fatty acid is attached with these PO4 and alcohol. Similarly, sphingosines is fatty acid, PO4 and choline. So, additional group is attached, so it comes under complex 1. So, phospholipid is major component found in the cell membrane. Now, we will go the internal structure of fatty acids. How fatty acid is classified? There are 2 ways. First is essential fatty acid, non-essential fatty acids. Essential means it cannot be synthesized in our body, that can be taken up through a diet. Non-essential fatty acids. Then another is classification based on carbon number. Okay. Long chain fatty acids, short chain, medium chain, very long chain fatty acids. So, another classification based on saturation, saturated fatty acid, unsaturated fatty acid. As I said, no double bond system found and double bond system found, depending upon fatty acid R. Now, how you will write If anyone asks you what mean by fatty acid, you cannot say that CH3, CH2, CCCOOH. Okay. Fatty acid. Acid means structure you will. Now COOH at the end, so therefore we are called as carboxylic acid with hydrocarbon chain ranging from 4 to 36. Fatty acids are two types, saturated, unsaturated fatty acid. Saturated fatty acid have no double bond system and thus the hydrocarbon chain is completely unbranched. Similarly, unsaturated fatty acids, these unsaturated fatty acids contain one or more than two double bond system, more double bond system usually found in cis configuration. Now PUFA, how you can say PUFA? PUFA means more than two double bonds or 2 to 6 double bond system. For example, I am giving you these unsaturated fatty acids is also classified or they are telling as MUFA and PUFA. Now what mean by MUFA? MUFA means mono unsaturated fatty acid. Another term is PUFA. PUFA means poly unsaturated fatty acids. Now what is the difference between? There are more than two double bond system. In MUFA, mono means single double bond system is there. What are the example? For example, mono unsaturated fatty acid, as I said, oleic acid. Okay. Similarly, PUFA for our brain, DHA and EPA. That is very much more importance. Linoleic acid, linolenic acid, 18:2. Linolenic, 18:3. This is PUFA. Okay. So similarly, how you see the structure of that? So hydrocarbon chain is there, one carboxyl group is there. So understand, you have to calculate. This is the copyrighted material that I will teach you later on. This 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Okay. COOH, this is also 18 carbons. So stearic acid are typical saturated fatty acids. You will not find any double bond between C and C, whether it is from here carboxyl end or CH3 end. So oleic acid are typical unsaturated fatty acids. Oleic acid, 18:1. Now delta position you have to remember from here sides, unsaturated fatty acids, C double bond C. 1, 2, 3, 4, 5, 6, 7, 8, 9. At delta 9 position you will find this. So MUFA is a oleic acid. Now how you will remember these classification of fatty acids? So you have to remember the LMP Sabel. LMP Sabel means lauric, myristic, palmitic, stearic, arachidinic acid, behenic acid and lignoceric acid. Now this saturated fatty acid in 12, 14, 16, 18, then 20, 22 or 24 symbolize with 12:0 means no double bond system. How you will remember this common name? LMP Sabel. Okay. Lauric, myristic, palmitic, stearic, arachidinic, behenic and lignoceric. Now one terminal, dodeca. Deca means 10. Dodeca means 12. Okay. Tetradeconic. Tetra means 4. Deca means 10. So 10 plus 4, 40. Similarly, octa. Octa means 8. Deca means 10. Octadecanoic acid 18:1. 18:0. Icosanoic acid. Ico. Here you have to remember sanoic acid. Ico. Docosanoic acid. So 22. 2 times. Similarly, tetracosanoic acid. So this cosanoic means 20. You have to remember. Similarly, How you will remember this? You have to, for example, if the structure asked in the examination, write down the dodecanoic acid structure, then how you will write? You know CH3, here is CH2, but in between CH2 means how n number you will calculate, -2. For example, first 12. So, write down CH3 into CH2, 10 and COOH. Okay. Similarly, you will find in the unsaturated fatty acid 16. 16 means palmitoic acid. Most of the oil that having a major proportion is 9-hexadecanoic acid. Here is 9 means ninth positions, hexa means 6 and decanoic means it has a double bond system. So, CH3, CH2, 5, then CH double bond CH and then CH, then CH2 COOH. But you have to remember 27 here. Similarly, oleic acid, linoleic acid, alpha linoleic acid, oleic, linoleic, alpha linoleic, gamma linoleic, all having 18 carbons, but remember the double bond system. Linoleic 18, alpha linoleic, that is also 18, gamma linoleic, that is also 18, but double bond system at 9-12, that is 18:2, that is linoleic acid. Alpha linoleic acid, that is at 9th position, 12th position and 15th position, octadeca trienoic acid. Tri means 3 double bond system. Similarly, 6, 9, 12, octadeca trienoic acids, that is also 18:3. So, one is 9, 12 and 15 position, here is 9, 12 and 6, 9 and 12 positions. Similarly, tetracosanoic acid, remember this, very important. Arachidonic acids 20:4, 24:1. So, in the examination we are asking what is the name of 16:1 fatty acids, then you have to name this is palmitoic acids. Similarly, 18:2 means linoleic acid. Generally, we are not asking alpha and gamma, but remember this fatty acid is very important for our brain and it is the precursor for DHA and EPA, docosahexaenoic acid, icosapentaenoic acid. These two fatty acids are essential for our memory as well as brain function. So, alpha linoleic acid, ALA, is very important. Similarly, 24 here also, 24 here also. Lignoceric is also found in many peanut species, but certain are problem with these fatty acids. Okay, I will teach you later on in PG also. Now, there are some dietary lipids, how you can classify it or where it is found and what is their proportions. The question arises in your mind whether these fatty acids or which source we will eat so that our body remain fit and we can survive for any damn environment. So, these are the edible oils and their composition based on the fatty acids. Now, you have to tell me palmitic, stearic, oleic, linoleic, lauric and myristic, LMP sub. Okay, that is LMP sub means saturated one. Now, oleic, linoleic and linolenic, generally these classification, most of the you will find 100% of that. So, in beef you will find oleic acid content from 37 to 43, or higher proportion is there. Similarly, in the coconut saturated fatty acid is higher, 74%. Okay, means higher lauric and myristic fatty acid found in what? Coconut. Similarly, higher oleic acid is found in which crop? Then you have to go through oleic, here you will find olive oil. Similarly, linoleic acid is found in which crop? Linolenic. linoleic. Tell me, yes, 78%. So, depending upon the mix up of this oil, you will find the proper proportion of the oil composition in your day to day food material. These are the, so in the, these are the beef and milk are animal origin. These coconut, corn, olive, palm oil, sunflower oil, safflower oil. Okay, these are the vegetable origin. So, how you can, as I said in the previous lecture, that wherever you will find essential, essential means it cannot be synthesized in human body. That has to be or given through a diet. So, definition of essential fatty acid that cannot be synthesized in human body must be taken in adequate amount in the diet. They are required for normal growth and metabolism. Which are these essential fatty acid? Linoleic acid, linolenic acid, arachidonic acids. These three are the essential fatty acids. Structural fats in the tissue which regulate the body functions such as blood coating, inflammation, and most of the fat soluble vitamins are also attached with that oil composition. So, essential fatty acid can result in the abnormality like poor growth, increased food intake, scale inflammation of the skin. Sometimes it also improved the immune system or immune responses. Besides these are the, as I said, dietary source of vegetable oils, corn oil, sunflower oil, peanut oil, and all rich in certain marine origin also there. So, oil rich fish, herring or sardine, these are the name of that fishes having a higher proportion of. Now, what is the function of essential fatty acid? Why, how we are taking these essential fatty acids? Whether they are useful in the treatment of atherosclerosis. Atherosclerosis is the medical terminologies. I will give you in depth what is atherogenic index, thrombogenic index. Okay, these are the certain lipid indices which will show you a deficiency disease caused by these essential fatty acids. They are useful in the treatment of atherosclerosis, essential fatty acid, arachidonic acid, linoleic acid, linolenic acids. Transport blood cholesterol and lowering it, transporting triglycerides. So, coronary heart disease are attached with this terminology. Okay. Hormones are synthesized from them. Many people are going to lower down the cholesterol. No, it is not essential. If you lower down the cholesterol level, that will lead to a deficiency or changes in the hormone content too. So, according to the ICMR or FDA also, they are now the level of this cholesterol must be maintained. It is simply they enter in the structure of all cellular and subcellular membranes, as I said. So, transporting plasma membrane phospholipids in the animal bodies, and they are essential for skin integrity, normal growth and reproduction. Why this in blue? Normal growth and reproduction. In many cases, you will not find a certain problems in the this terming. Okay, because of cholesterol synthesis, and they are lowering down their weight, and that will leads to this deficiency. They are an important role in blood clotting, intrinsic blood clot, thrombogenesis. So, these important in preventing and treating the fatty livers, and important role in the health of this retina and vision. That I will teach you in the vitamins also, vitamin A, D, E, K. These are the fat soluble vitamins, and they are essential for this. Okay, they can be oxidized for energy production. So, these are the essential fatty acids, and it has a double bond systems. it also have properties of oxidations. So you have to remember that can also be asked in the examination, what mean by essential fatty acid, describe their functions and properties too. So I will give you a physical properties of fatty acids. First is physical property of fatty acids are determined by the length and degree of unsaturation. What mean by degree of unsaturation? Double bond system found in that, okay, hydrocarbon chain. Longer chain or fewer the double bond. Longer chain, CH3, CH3, CH2. Longer chain and fewer the double bond system, lower is the solubility in water. This is the universal sentence. Lower in the solubility in water and higher in the melting point. Melting point in dissolved. Addition of double bond decreasing the melting point, whereas increasing the chain length, increasing the melting point. These are the universal sentences that can be asked in the multiple choice question. So remember these words: melting point, decreasing the melting point. How you can decreasing the melting point? Addition of double bond. Similarly, fewer the double bond means saturation is more, then it has a higher the melting point. So trivium of fatty acid refers to a natural source of derivations that palmitic. Why this name came? As I said, lipos means fat. Here fatty acid name. For example, peanut, arachidonic acid, palm oil, palmitic acid. Similarly, oleic, that is from oleic acid, olive oil. So palmitic acid 16:1 seed fat of palm tree. Oleic acid, olive oil. Okay. Similarly, now physical properties of fat and oil. Freshly prepared fat and oil are colorless, odorless, tasteless. But this is not for universal. Certain oil are yellow in color because of pigments is found in that during the extraction procedure. So any color or taste is due to the association with the foreign substances. Yellow color of the body fat or milk fat is due to, as I said, pigments. If it is cow milk, for example, cow milk or cow ghee, you can see it can differentiate. You will find yellow color. Here you will not find yellow color. That is due to keratin pigments. Similarly, but fat have a specific gravity, less than 1. Therefore, it float on water. Fats are insoluble in water. Why? As I said previously, but soluble in organic solvent, more double bond system. Melting point of fats are usually low but higher than the solidification point. So one by one we will see the chemical or properties of these chemical properties of fat and oils. Hydrolysis means water is involved. So remember this word, hydrolysis. They are hydrolyzed. They are means lipid. This triacylglycerols. Lipase is the enzyme. Lipase is 3H2O remove here. R1, R2, R chain, these are the fatty acid attached with this. So they are the hydrolyzed into a fatty acid and glycerol. Here is free fatty acid and glycerol. Wherever you will find free fatty acids, more free fatty acids means it degrade very fast. Okay. So by the action of superheated steam, acid, alkali or enzyme that is lipase of the pancreas, during their enzymatic and acid hydrolysis, glycerol and free fatty acid is formed. Okay, first properties. Now another property, saponification. Mostly you will find soap. This soap is made up of this process, saponification. Now how you will understand this saponification? What is the definition of that? milligram of KOH or NOH required to saponify 1 gram of fat or oil. That is called saponification number or saponification value. Now here it requires NaOH or KOH. Means alkaline hydrolysis produces the glycerol and salt of free fatty acids. These salt of fatty acid, that is called SOPs. Now SOP cause emulsification of oily material that help easy washing of fatty materials. So dust can remove or any dark spot found in that. So it require alkali. Alkali means NaOH, KOH. Here 3 NaOH, triacylglycerol, glycerol and sodium salt of fatty acid. So each at R1, here is R1, R2, R3. COONA, Na 3 times. Similar structure, similar things. Simply 3 NaOH. In hydrolysis 3 H2O. Now halogenation. Halogenation means neutral fat containing unsaturated fatty acid have a ability to adding halogens. What are the halogens? Simply hydrogen or hydrogenations, iodine or iodination at the double bond system. It is attached there. It is very important property of determine the degree of unsaturation. So every property is related with the certain terminology. So you have to remember halogenation, that is determine the unsaturation of the fat. Degree of unsaturation, that already been taught to you, that determine its biological values. Here is example of stearate tetraiodate is attached with CH positions from here or here too. So we will go fast. Hydrogenation or hardening of oil. Yes, this property is very important. Nowadays you cannot provide fat only through the animal origins. Jisko vegetable ghee. Okay, this vegetable ghee is produced through this. It is the type of addition reaction accepting hydrogen at the double bond of unsaturated fatty acid. That is done under the high degree, high pressure of hydrogen, and that is catalyzed through nickel or copper. Nickel and copper is the catalyst, and during heat process that will hardening process of oil. Simply, very simple method. It is the base of hardening of oil. Margarine, manufacturing, changes of oleic acid of fat into a stearic. Remember, oleic acid of fats into a stearic of stearic acid, it means what? Liquid convert into a solid, means halogenation, hydrogenation. Hardening process is that it is advisable not to saturate all double bond, otherwise margarine produce will be very hard, of very low biological value and difficult to digest. Therefore, you have to use this oil or liquid with unsaturated fatty acid only convert into a stearic, that hard fat margarine. Now, what are the advantages? Advantages of this hydrogenated oil. It is more pleasant as cooking fat. So, in bakery also certain are giving this oil. It is digestible and utilizable. As a normal animal fats, it is less liable to cause gastric or intestinal irritations. It is easily stored and transported, less liable to rancidity. You will not find any bad odor during that. So disadvantages also there, because of most of the fat soluble vitamin A, D, E, K. Fat include lack of fat soluble vitamins and essential fatty acids. These are the disadvantages and advantages of hydrogenated oil. Now rancidity. This topic you have to understand very well. Why? Rancid means bad odor. This bad odor can be possible with all living organisms. Wherever you will find deadly materials, the bad smell come out. If the rat killing here, you will find a bad smell because of this rancidity. So remember this, rancidity means what? Bad odor. How? These are the two types of rancidity, hydrolytic rancidity and oxidative rancidity. I will give you a later on. When butter or fat is stored for a long time, they often become a rancid. Why? Because of two reasons, hydrolysis or oxidations. The rancid fat gives a flavor, bad odor, bad smell, unpleasant taste also. The rancidity can be defined. Now that can also be asked in the examination. Describe types of rancidity and define rancidity. So define rancidity means a production of bad unpleasant odor or smell in the oil or a fat is called rancidity. Rancidity of two types, as I said, hydrolytic rancidity, oxidative rancidity. Oxidative rancidity is also classified into two. One is aldehydic, another is ketonic rancidity. Now hydrolytic rancidity, that is developed by the action of microbial lipase, attack of microbes on that enzyme which split triglyceride and produce low molecular weight compound, low molecular weight free fatty acids, C4 to C10, that such as butyric acid. This butyric acid produce bad smell in the butter. Butter or butter fat plus enzyme is lipase. If your water is there, then it will convert into a glycerol plus butyric acid that produce an unpleasant smell. That another is oxidative rancidity. This oxidative rancidity developed due to the oxidation of unsaturated fatty acid to produce either cleavage or polymerization of fatty acids, or there are two types, aldehydic rancidity, another is ketonic rancidity, CHO, C double bond O. These aldehydic rancidity, this production of peroxide, H2O2. When the peroxides are converted into aldehyde, peroxide converted into aldehyde, which gives a bad smell in the fat. Similarly, ketonic rancidity. When ketones are formed, how? Beta oxidation of the saturated fatty acid, then keto acid are formed. Elimination of CO2, that will lead to a rancid ketone formation to produce bad smell. So this is all about the rancidity, that can also be asked in the example. Now hazard or hazards of rancid fats, as only they are not producing a bad or unpleasant smell, they will give you a bad taste too. Similarly, they are producing certain toxin too. I will give you a 4 points. Why it is hazardous? Because the product of rancidity are toxic, causes food poisoning, and sometimes it also causes a cancer too. Another is rancidity destroys the fat soluble vitamin A, D, E, K. Okay. Rancidity destroyed most of the PUFA, which is essential for formation of other fatty acid, essential fatty acid. So rancidity destroys the PUFA. Rancidity causes economic losses too. If oil is destroyed or storage oil is become rancid, then economic losses is certainly there. So it become inedibles. Now another how it can prevent. Rancidity can be prevented through the addition of chemicals, that is called as food additives, that I will give you later on, and how you can avoid this. There are certain technology is there. Evidence of this cause is exposure to light, oxygen, moisture, high temperature, and certainly during processing bacterial and fungal infection, contamination should not be there, so that you can preserve that oil in well closed condition. Then simply you have to put into a dark place. dry place, cold press, good storage condition should be maintained. Removal of catalysis, lead, copper that catalyze rancidity. That is not possible for everything. Addition of antioxidant. Yes, that can be possible to prevent the peroxidation in the fat or oil. They include phenol, naphthol, tannins, hydroquinones. Most common natural antioxidants is vitamin E. In many collectors, they are giving in certain areas you will find this vitamin E addition in the most of the oil. Why they are adding? There are a certain permissible limit too. So vitamin E and certain vitamin is also addition for food fortifications too. Now, qualitative test or analytical constant to determine the purity of oil and fats. There are certain parameters, chemical parameters that can determines the oil and fat, or it is very essential to keep the oil in pure hygienic conditions, and certain tests should be for the malpractices or adulterations also. So malpractices by what? Who are doing these? Generally malpractices is being done by these oil millers, and there is the chances of adulteration of purity of oil too. So there are analytical constant given by judging the quality of oils. They are also given by the Food Safety and Standard Authority of India, Food and Drugs, and Codex standard is also applied for the various oil from transporting from India to other countries too. So fat constant or number are tests used for checking the purity of fat and detection of adulterations. To qualitatively estimate certain properties of fat, to identify the biological value and natural characteristics of the fat, detection of the fat transit and presence of toxic hydrolytic fatty acids. These are the certain objective behind to understand the fatty acids. Now one by one I am giving you a chemical properties only. So you have to understand what is iodine number, what is saponification number, what is halogenation, what is RAM value reached. One by one I am giving you a completing this definition of the iodine numbers. Means it is the number of grams of iodine absorbed by the 100 grams of fat or oil. That uses why we are measuring iodine numbers, because it is measured the degree of unsaturation from the fats or natural properties of the oil. Unsaturated fatty acid absorb the iodine at their double bond systems. Therefore, the degree of unsaturation that can be more than iodine absorption, the degree of unsaturation increase, the iodine number. Hence, the biological value of fat is also increased. It is used for identification of type of fats, detection of adulterations, determine the biological value of fats. This parameters is also taking saponification number that already been taught you. It is the number of milligram of KOH required to saponify 1 gram of fat or oil, or these are they also useful for what? What is the chemistry behind that? Then since each carboxyl group of fatty acid react with this 1 moles of KOH during the saponification process, therefore the amount of alkali needed to saponify weight of the fats depend upon the number of fatty acids. Thus, the fat containing the short chain acid will have a more carboxyl group per gram than the long chain fatty acid and consume more alkalis. This is the saponification. Now, acid numbers, it is the number of milligram of KOH. Similar words, you have to remember only a neutralize. So, it is the number of milligram of KOH required to neutralize the free fatty acid present in 1 gram of fat. That is called acid numbers. Uses it is for detection of hydrolytic rancidity. Remember these words because it measures the amount of free fatty acid in oil. So, acid value or free fatty acids, it is very important for determining the quality of oil, whether or storage of the oil. The lower the fatty acid value means oil or fat fresh. Okay. Similarly, these RM numbers or Reicher's-Michels numbers or a value that you have to remember. Definition of this, it is the number of ml of 0.1 normal KOH required to neutralize the water soluble free fatty acid. Fatty acid distilled from 5 gram of fat. Short chain fatty acid less than 10 carbons, it is distilled by the steam, and this neutral fat, natural composition of this fat is useful for detection of fat adulterations. Now, butter has a high percentage of short chain, has a higher RM number compared to margarine. Now, P number means Polensek number. P number is also, it can be defined as number of ml of 0.1 normal alkali that requires to neutralize water soluble. These water insoluble and volatile fatty acid containing 5 gram, that is also from 5 grams of fat or oil. P value decreases or increasing beyond the range, there is a chance of adulteration. So, RM number and P value is very important for determining any ghee is adulterated with, or that oil is adulterated with the other things. This K value is nowadays it is not found, but you have to now, not Krishna, you have to remember the pronunciation is there. This Krishna value, K value, SCH, Krishna value, it can be defined as number of, same word is there, 0.1 normal, but here is water soluble. 0.1 normal KOH alkali required to neutralize water soluble and volatile fatty acid contained in the 5 grams of fat or oil. K value decreases or increases beyond the range, there is a chance of adulteration, or the quantity of butyric acid can also be measured through this. Now, in certain examination, I will tell you what is the difference between fat and oil than ester of fatty acid in glycerol. It is also ester of fatty acid and glycerol. Solid at room temperature, oil is liquid at room temperature, lower melting point, higher molecular weight, more sustainable or saturated fatty acid found in the fat, higher unsaturated fatty acid there, higher saponification value, lower saponification value is there in the oil, low iodine value, whereas in pound of oils high iodine value. So, this is all differences of fat and oils. Now, acetylene number. This acetylene number or value, it is the number of milligram of KOH needed to neutralize the fatty acids or acetic acid liberated from these hydrolysis of 1 gram of fat or acetylated fat. So, natural or rancid fat also contain fatty acid with the free hydroxyl group, that is also converted into thus acetylene number is also a number of hydroxyl group present in, and it is used for studying the natural properties of fat or adulteration of also. These are the functions of lipid. As I said, lipids are constituent of cell membrane, insulating material, protect the internal organs. It is also found in the fat soluble vitamins. Removal of phospholipid help in the removal of cholesterol from the body, and phospholipid in the mitochondria, that is also responsible for the transport of electron transport chain component. Similarly, lipid cholesterol is one of the major component or constituent of any membrane structure, and it is synthesized for very important things, that is hormones and vitamin D. So, it is the principal sterols found in higher animals. As a storage form, lipid provides 9 kilocalorie per grams of that. Their main function is provide essential fatty acids. This is a repetition, synthesis of hormones, that is also repetition, adrenal cortic and vitamin D3, heat insulator, presence of fat is subcutaneous tissues provide insulations. The well is it is also insulation animals in Antarctic regions. As I said in the previous pictures, mechanical protection, it gives you cushions such as heart, brains, very smoothie. They are used in the perfumes and flavor industries also. It reduces the bulk of diet. Some essential fatty acids also make unpalatable or aroma or attractions of the certain animals or insect. That can also have properties of lipid. I will tell you in the volatile oil compositions. These are the importance of glycerides in the everyday lives. Hydrogenation, saponification, drying oils, wax, castor oils in the medicine, cosmetic industries, toilet industries, soap industries, paint industry, waxes. These waxes are long chains 14 to 36, and properties of waxes are insoluble in water, soluble in waxes are not easily hydrolyzable as a fat. indigestible by the lipase. These are the example. You can see there are no nutritional value or function of wax, bee wax, carbon wax, permittive wax, or octacosonelic wax. These are the various sources of that. Wider application you will find in the pharmaceutical industries and cosmetic industries, and protect the skin and hair. So the wax's cuticle layer is found also in the plant tissues. Therefore, the insect cannot survive, or water is also flow downs, and many compound lipid I will tell you later on. Thank you all. Thank you. This topic is very